Hi everyone, welcome to another episode of Science and Technology Q&A for kids and others. I'm going to try and answer questions that you guys give me about, well, anything you want in science and technology. I'll do my best. I see here a question that came in on our submission form from Sam. How was it possible for civilizations across the world to both develop pyramids independently? Egypt, the Mayans, Aztecs, etc. Is there scientific significance to this? Interesting question. In both technology and biology, there is occasionally convergent evolution. Like, for example, the eye, taking light, turning it into nerve impulses, electrical signals in nerves. The eye has been invented multiple times in biological history. And each time it's been invented, like for example, we have eyes that have a particular arrangement of cells on the retina, compared on the back of the eye, we actually, our eyes got it wrong. We have some cross connections in front of our retinas, so that all the photons of light have to go through the cross connections before they get to the light sensitive cells on the back of our retina. Things like, I think, king crabs evolved eyes independently and got that right. So there are cases in both biology and technology where things get invented sort of independently and end up being sort of functionally the same. And I suspect that's what happened with pyramids, because I suspect that it was kind of like, well, you can arrange things in a particular way, and this is a way so that the stones don't fall down and it looks cool. And so it got independently invented. There are definitely some mysteries in terms of independent invention. Like, one of the ones I've been curious about is kind of labyrinth shapes, kind of maze-like shapes. There's a particular maze shape that you see on coins from Crete, for example, and from, I don't know, 4th, 5th century BC, things like that. And it's a particular pattern, but that same pattern shows up in South America. It shows up in other places in Europe. And I found that quite mysterious, because it's a pattern where it doesn't really seem to be that distinguished from other patterns. It seems like you could have invented a bunch of different maze patterns, but that particular maze pattern shows up in those different places. And it kind of eventually tells one, well, there was presumably some kind of communication, because it's sort of a random thing that showed up. It's not like a pyramid where you start piling rocks up and there's sort of it feels almost inevitable that you'll end up inventing the idea of a pyramid. This seems a bit more specific. That particular maze pattern has a funny history. It's has some kind of mythical character. It's sometimes Daedalus is said to have been the architect of the labyrinth of the Minotaur. The Minotaur was some kind of bull composite monster creature that lived in the sort of it lived in the basement of the palace on Crete, or at least is said to have done so. And apparently that the myth goes that Daedalus, famous chap from antiquity, mythical chap from antiquity, designed that labyrinth. And then I think there's also a statement that it was the logo of the city of Troy, or maybe it was the design of the city walls of the city of Troy. But anyway, a particular pattern that just shows up in a bunch of different places. There are other things where you could say, how convergent was the evolution? How divergent? Another one is like numbers. The idea of numbers got invented multiple times. The idea of writing got invented at least 3 times. But each time writing got invented, each time numbers got invented, they were configured a bit differently. Like, for example, the tradition from ancient Babylon, cuneiform, the Phoenicians, leading to our alphabet and so on. That was sort of a very alphabetic system, whereas, for example, the Chinese system is ideographic. Characters represent whole words effectively. Different invention, different construction for language. There are some features of language that are sort of curiously convergent. Like, for example, if you look at the languages of the earth, there are maybe 7,000 languages that are still in use today. Unfortunately, languages can go extinct when, you know, a language is only really fun to use if multiple people talk it. If you're the last speaker of some particular language, it's tough to justify continuing to use it. It's not a good form of communication because there aren't other people you can communicate with in that language. And so there's a certain rate, unfortunately, in modern times of languages going extinct. But I think there are about 7,000 still left. And one can ask kind of what commonalities are there, what convergence of evolution, what divergence of evolution are there in those languages? And sometimes you can see there are definite families of languages. I mean, English, for example, is part of the Indo-European family of languages, but there are many other families of languages. Occasionally, there are languages which are isolates. Languages like, I believe, Basque, a language in northern Spain, is an example of an isolate that is a language where there's just nothing else quite like it anywhere. There are other languages like for example, Finnish-Hungarian family of languages that are different from the other European languages, but are at least in a group by themselves. But, you know, so you can kind of trace the sort of evolution of languages, but there are certain things which seem to sort of always be there, like nouns and verbs and adjectives and so on. Those are always there, it seems, in every human language. And those are places where, in a sense, the presence of those things probably has something to do with the way that sort of our brains are set up and the way that we tend to think about things. You know, I'm always very curious about the structure of languages and the extent to which one needs a particular form. And in designing computational language, I've certainly leveraged the fact that people are so familiar with human languages which have certain characteristics. You know, verbs tend to be very much like functions and programs and so on. Nouns are like entities and literals and things like this. And it's useful to kind of leverage that understanding. But in terms of, I'm curious, you know, if one looks at sort of the evolution of technology in the world, you know, ideas like wheels or fire, you know, were these invented multiple times, or did they have one inventor, and did these things then spread? I think different kinds of things will have had different levels of one inventorness. The thing that I always find interesting is there are more things than you might think that just had one inventor, where it wasn't obvious it was going to happen ever, and somebody somewhere invented it. I think that I mean, it's probably worth remembering that in antiquity, there were many fewer people around in the world than there are today. And so many fewer sort of places for invention, although sometimes invention is easier when there are fewer people around, because when there are lots of people around, there's a certain collective belief that, oh, no, you can't invent that. That's not, you know, we all know it works this way. Whereas if there are fewer people, there's less kind of a herd mentality of what you have to do. But it is certainly, it's often there are things where sort of they finally got invented, and you say, why was this never invented before? It's surprising that it wasn't. I mean, there are things that I've been lucky enough to discover in my life in science and so on, where you look at them, particularly things that involve, you know, simple rules, and just apply the rule and you see remarkable things that happen. You look at them and you say, this could have been invented 2,000 years ago. How come it wasn't? And okay, you feel good about the fact that, well, I managed to invent it. Usually the real reason why one managed to invent it, and it took a couple of thousand years, is because there's some sort of overall context. There's some set of other things that one understands that make it worthwhile to think about those kinds of things, and that hadn't existed in the past. But it is always interesting to see invented sort of as a result of a particular person doing things. There are other things where occasionally the world gets to a certain point and then the same thing is invented because there's sort of ambient knowledge in the world. The same thing is invented 3 times sort of in the same year just because that's kind of the stage that the world has reached. Yeah, so I don't know specifically about pyramids. I don't know. Let's see. Well, let's look here. Gosh. It's a question from Dougie. Does the double helix shape in DNA show up anywhere else in nature? Interesting question. You know, the primary feature of, you know, DNA has this helical form. I mean, that's its most obvious feature. But then, you know, an actual piece of DNA, like human DNA, is a meter long, but it's in every one of our cells. So it certainly isn't that long, thin thing that's just coiled around in a spiral, you know, helix type shape. It's all curled up in a tertiary structure that has, which has all kinds of pieces, and there are all kinds of issues about whether it gets knotted and so on. And quite likely some of the functionality, that some of it is known, but some of the functionality of the DNA actually depends on the way in which it gets curled up like that. So, I mean, this idea of kind of the double helix and the concept. So just to explain, DNA, it's the molecule that has the surprising feature that one molecule can store lots of information. And that's how we pass our genetic information on. And there is a copy of our DNA in every cell in our bodies. And DNA has the way it works is there are these clumps, these bases that are clumps of, I don't know how many, but about 20 atoms, 15, 20 atoms. And there are 4 kinds, A, T, C, and G. And they, along the backbone of DNA, you have the sequence, you know, A, T, T, C, G, G, whatever it is, some long sequence. For us humans, there are altogether 6 billion of those base pairs that encode the way that we should build proteins and create molecules that make us up. But in any case, the thing that is the case is that the chemistry of DNA is such that you can kind of switch in any of those A, T, C, and G on this backbone, and the kind of, you can make a molecule in which you can have any sequence there. Most molecules, you know, you'll have a particular set of atoms in that molecule, and that's the way it'll be. You don't get to kind of mix and match clumps of atoms in the molecule. There are other sort of a simpler version of something like DNA is polymer molecules like PVC, polyvinyl chloride, or I think nylon is an example of this also. Molecules where there's a single unit, a single collection of atoms, and just repeats over and over again. And that gives you this long stringy molecule. And that's an ordinary polymer. DNA is sort of technically referred to as a heteropolymer. It's a polymer where there's repeating units. It doesn't have to be the same unit repeating many times. Instead, it can be these 4 different possible units, and they can repeat kind of in any order one chooses. And what that sequence is where the information, where the genetic information is stored. Now, DNA is this double helix because, well, the fact that it forms a helix is a feature of the backbone of the DNA molecule, and you're probing pieces of chemistry I do not know well. I think it's a phosphate. I think phosphorus is somehow involved in the backbone, and I'm sorry to say I can't immediately tell you how that works. What I can tell you is that there's a strand. DNA always has 2 strands, and if you have an A on one strand, there's always a T on the complementary strand. If there's a C on one strand, there's always a G on the complementary strand. And so DNA essentially has these 2, a positive and a negative copy of a single DNA molecule has a sort of positive and negative copy of your genetic information. And the trick that DNA uses and that life uses is that you can essentially copy a DNA molecule by having those two strands unzip from each other, so they become something where there's just an A and it doesn't have a pairing up T on the other side. And then that single strand can then be refilled with a second strand, complementary strand, that then has, for every A, it has a T, for every G, it has a C, and so on. And that's how replication happens, is that the DNA unzips during cell division, it unzips and then refills its complementary strand from material that's provided in the environment, so to speak. And so that's the sense in which it is a double helix, so to speak. I think the helical shape is not probably the most significant feature, and I'm not sure exactly what functional significance it has. I think it just is a helix just because the way that the backbone is set up is such that it just doesn't make a straight backbone, just by the nature of the atoms that form that backbone. I don't think there's any great significance to that. When you deal with DNA, there's always the 3' direction and the 5' direction, which describe the sort of the outer side and the inner side of the helix. And if you I myself got my whole genome sequenced back in 2010. Actually, I've had it sequenced several times since then, just because I'm a collector of data. But when I'm curious whether some newly published finding about genetics, whether I have some variant of some particular gene or not, I'll go and I'll look at my genome, and it's just a series of, in Morphin language, it's just a string of characters, A, C, G, T, et cetera, et cetera, et cetera. And you always have to, it's always a little tricky to go and see, somebody publishes a paper that says, if you've got ATTCG in this section of this particular gene, then you'll have brown eyes or something. Then to recognize, to find out what part of your genome actually that is. Because see, the thing is, everybody's genome is slightly different. There's 6 billion base pairs altogether, but we all have many millions of base pairs that differ between different people. And sometimes there'll be additions to the genome, sometimes there'll be subtractions. So you can't just say this particular at position 3,700,417, there should be an A. You can't really say that, because somewhere upstream of that, you may very well have all kinds of insertions and deletions relative to kind of the default standard genome that exists. So you have to be a little bit trickier to look at flanking sequences of, I have this, this sequence of letters only occurs at this place on the genome, so I kind of know where this is going to be, and so on. And it's always tricky to get the right side of the double helix. Now, this question about whether there are other self-replicating molecules, well, we don't know of any others. You can have sort of modified versions of DNA and so on, but in terms of completely different chemistry to make a self-replicating molecule, there aren't examples known. There've got to be examples. It's just we haven't been quite imaginative enough yet in chemistry to have come up with them. Now, people wonder, in the origins of life on Earth, it's pretty remarkable we ended up with this molecule that so successfully can replicate itself. And people kind of wonder how it came to be that way. And I think the general, a common belief is that in things like clays, so a clay is something where there's sort of a kind of a matrix of mud, and there's kind of other minerals that are in that clay. And one can imagine a situation in which there's sort of a solid template that creates molecules. So with DNA, what ultimately happens with the DNA, it gets the RNA makes copies of what's on the DNA, and then the RNA is used as a template to specify, essentially unit by unit, amino acid by amino acid, how to make a protein. And we are made from, 30, 50,000 different kinds of proteins, which are specified by our DNA. But so for us, we are making molecules to order by having on the ribosomes in our cells take in the RNA that is transcribed from the DNA is specifying the sequence of units, the sequence of amino acids that will be extruded from the ribosome to make proteins. So our proteins are just sequences of these amino acids. So the question is there any other way that you can, in a templated fashion, make molecules? And the most obvious kind of thing to do is to have a solid template, a template which is at a molecular scale, is going to say, you know, put this atom here, put that atom there. And what the way that the atoms get put there is, well, the atoms are floating around in a liquid or something like that. And if that solid template has a particular form, has particular electrical properties or something at that particular place, then, an atom of this type or a radical, a group of atoms will tend to bind, will tend to be attracted to that place. Another one will be attracted to another place and so on. And one can imagine a situation in which one would kind of be making a molecule on a template on something like the clay. Now, you know, are molecules made on templates? Well, yes, they are. That's actually a lot of how biology works and how also a lot of industrial chemistry works. In catalysts, so normally you have a chemical reaction. What is a chemical reaction? You have a bunch of molecules, they're all floating around like in a liquid. Most chemical reactions take place in the liquid phase because that's where molecules are fairly dense, unlike a gas where they're much more less dense. In liquid, they're dense, but they're just always bouncing around randomly. And most of the time when molecules bounce around in liquid, they just bounce off each other. But if there is the potential for a chemical reaction, then occasionally when two molecules hit each other, they'll be arranged in just the right orientation that the molecules will exchange electrons, they'll change their configuration, they'll interact, they'll produce a new molecule. And that's what a chemical reaction is molecules colliding, they get close enough in the right orientation so that they can have electrical, or they can sort of exchange electrons to change the structure of bonds between the atoms and so on. Okay, so that can happen just by two molecules just randomly crashing into each other. But the other possibility is that the first atom can be, for example, can sit inside some place on a surface, some particular sort of patterning of some surface at the molecular scale can sort of have a molecule just sit in that hole, let's say, and then another molecule will come along and will fit itself into that first molecule. And so that's how catalysts tend to work. That a catalyst, the most common case, is that it's some solid, and the surface of that solid has a certain roughness or certain features which cause the molecules to kind of they get attracted to the surface. They sit on the surface, and they are by the features on the surface, those molecules are oriented in a particular way, and oriented in just the right way, so that when the molecules that are sort of floating around on the liquid or something come there, they will be more likely to sort of attach in the right way and produce a chemical reaction. So that's what happens in lots of industrial chemistry. And it's what happens, I don't know, catalytic converters, for example. It's pretty common to use metals like platinum and so on, because they have certain features of their surface. Their crystal structure leads to certain features of their surface which are convenient for kind of aligning molecules the right way. The other case that happens is in biology, where enzymes are molecules that sort of catalyze reactions. They're typically big protein molecules, and they'll have a certain shape, and the shape will have certain holes in it, certain valleys, certain peaks, and so on. And what will tend to happen is that one molecule will kind of be attached to that enzyme in that particular way, and then can set things up so that it is more likely that a reaction will happen. And often the rate of the change in the rate of reactions associated with catalysts or enzymes or whatever is huge. It's factors of a billion and so on. It really matters that these molecules aren't sort of just bouncing around randomly in random orientations, but they're actually sort of arranged in the right configuration to undergo a chemical reaction. So let's see. So question here. Oh, I was going to maybe just finish on this question about molecules that can copy themselves. I think that this feature, I mean, it seems easier to kind of arrange this to happen at the level of a molecule imprints itself on a solid and kind of patterns the solid in a certain way, and then something else comes along and uses that template in the solid to make another copy. It seems a bit easier than the case with DNA, where it's kind of molecule to molecule type stuff, but one doesn't know. And it's interesting to kind of imagine if we were going to redo life, how would we set it up? And for example, as we start thinking about molecular computing, it's of great interest to try and see how we replicate some of the kind of methods that are used by living systems to kind of do things with molecules. Which leads us to a question here from Erin. Are there any examples of logical gates being built out of chemical reactions? What breakthroughs are needed to achieve this? How many gates are needed for a programming language like C? Okay, so yes, people have made logical gates out of chemical reactions, though usually they've used DNA to do it, which is kind of a you know, you're building a giant tower to achieve something comparatively simple. The most common case is that they'll use DNA, they'll use the fact that DNA has this matching property to make kind of tiles out of DNA, so that, which have the property that they only match in certain configuration, and therefore you can arrange for sort of to make a progressive jigsaw puzzle that will do things like corresponds to a logical gate. Now, I have to say, I think that's an unbelievably overcomplicated way to do it. And in fact, some things I'm looking at right now might lead one to much simpler ways to make the equivalent of logical gates out of molecules and interactions between molecules. In fact, just yesterday, we were doing a live stream about molecular computing and subchemistry, where we're talking about some things which might be a run-up to that. We didn't talk about logical gates, actually. Maybe we should have done. Just to explain to people what logical gates are, when this is kind of an idea from the origination of logic back in antiquity, but it sort of was cleaned up in the 1800s, and then cleaned up even more when electronics became popular. But the idea is you've got P and Q, and you say P and Q. What is P and Q? It means if both P is true and Q is true, then the result P and Q is also true. Or you can say P or Q. If either P or Q is true, then the result is true, otherwise it's false. And so when you go to, well, it used to be the case, when you go to an electronics store, you can buy an AND gate, so to speak. You know, I don't think that's really a thing anymore, because these days there are microprocessors that have, you know, 1 billion of these kinds of gates sort of already printed on them, and you don't really get the individual AND gate type thing. But you can make, you can arrange sort of combinations of those gates to represent any kind of logical expression. So, and by having sort of, by having memory and feeding things back and so on, you can use that to make any computation you want. Now, the thing that was discovered about 110 years ago now is that all these different logical gates, you know, AND gates and OR gates and NOT gates and exclusive OR gates and so on, all of those gates can be made from combinations of just one kind of gate, a NAND gate. So what does a NAND gate do? If you have P NAND Q, it's the NOT of P and Q. So for example, NAND, let's say P and Q are both true. Okay, so P and Q is true, but NAND, P NAND Q is false. So if true is 1 and false is 0, so that means that 1, 1 will be 0. 1, 1 going into a NAND gate will give an output of 0. 1, 0 going into a NAND gate will give an output of 1, and so on. But so it turns out to be possible to make anything out of combinations of NAND gates, any logic operation out of combinations of NAND gates. Like, well, let's see, what is, oh my gosh, I should be able to do this. NAND of P and P, I think, is not P. And you can keep going, you know, NAND of P of NAND of Q and R and so on will make an and so on. Some of them are kind of complicated, but you can make anything out of a NAND gate. So if you can make a NAND gate from chemistry, then you can make any logic operation from chemistry. And it's a good question whether there's an easy way to do that by just looking at chemical reactions. Good thing for us to look at, actually. If you want to make kind of a programming language, well, you actually don't need, you need memory. You need the ability to store bits of information, but you don't actually need a lot of structure in the gates that you have. I mean, a typical microprocessor these days might have 1 billion NAND gates on it, but in principle, you don't need all that many NAND gates. You're at the cost of having a more complicated program, having more complicated software, you can have a vastly simpler setup. Like, for example, well, I had this idea years ago that of the very simplest possible Turing machine. A Turing machine is kind of idealization of computation that's based on, you can base it on logic operations if you want to, logic operations plus memory. Turns out you can have a Turing machine with just 6 sort of cases for its logic that is universal, is capable of any computation you want. And that's the very simplest possibility, but it's really simple. And as something that might be made of molecules, it's really quite a plausible thing. And actually, we didn't talk about that in our discussion of molecular computing yesterday, and we should have done. Anyway, so let's see. All right, let's see. Baker asks, is it necessary to have supercomputers to do meaningful biomolecule level simulations? So the question is, you've got a molecule. It's made of a bunch of atoms. You can usually, you know, people often draw these kind of ball and stick diagrams representing molecules. They're an approximation. They're not perfect. In reality, a slightly better model would be to say they're these atoms and they're connected by kind of springs to other atoms. And so if you take that molecule and you kind of kick it, it will vibrate around, and one can actually measure the vibrations of molecules. Is an important thing to do in recognizing molecules in spectroscopy and so on. Even that's just an approximation. The true story is a whole bunch of fancy quantum mechanics and a bunch of fancy mathematics. It's super difficult if you go to the level of that fancy mathematics to work out, given a certain set of atoms, what will the configuration of the molecule made of that atoms be? That's really difficult. The limit of what people can do is maybe 100 or 200 atoms. To be able to do that kind of calculation from scratch, using all the fancy mathematics of quantum mechanics, and say, given these atoms, what will a molecule made of those atoms be like? So usually one's dealing with approximations, when one's dealing with the kinds of things that happen with millions or billions of atoms that might, well, millions of atoms that might exist in a protein, for example. One is not able to do things at the level of the perfect mathematics and quantum mechanics that apply to sort of individual atoms. So one's using an approximation. One's using an approximation where one says that the force between atoms is governed by a certain force law. But even with that approximation, it's still difficult to work out what this 1 million atom thing will do. And a long-running issue is when you have proteins, which are made from millions of atoms, how will the proteins fold up? The proteins are made in a line. They come off the transcription of the DNA sequence, and there is a line of amino acids. But proteins, in practice, most proteins fold up. Some proteins don't. Some proteins, like the ones in our muscles, are long and stringy, but many proteins fold themselves up in this kind of globular structure. And it's been a long time issue to figure out how that folding works, even though the kind of in principle, we kind of know the physics of what causes that folding. We know it at the level of quantum mechanics, that's super difficult. We know it at the level of force laws, that's just difficult. And for a long time, it was sort of an unsolved computational problem. How does a protein fold up? There were typically two schools of thought. One is you just compute the forces and you see how the thing would actually fold. The other is you just look at how other proteins folded, you try and extrapolate. You kind of just try and use pattern matching effectively to say, I've seen a protein that looks like this and it folded like that. What's happened in the last couple of years is that with machine learning methods, pretty much we'll see how the other proteins folded method has won out. And that's now allowing one to do pretty good protein folding calculations. Probably what's really happening there, though, is that there are particular patterns of folding that we've never really identified, but which the computer is successfully identifying. We do know that proteins will often fold up in helices, actually, alpha helices they're called, or beta sheets. But there are probably some other motifs into which they fold that are the real things that these machine learning methods are keying into to figure out sort of what the overall structure of the protein should be. But to know how the protein will behave, like if you kick the protein a certain way, how will it blob around? What will it do? Or if some other molecule comes close to the protein and starts pushing pieces of it away, and maybe there's an active site that opens up and things bind to it, to know how that all works. That's really hard. If you look at simulations of molecules, there's a nice, our former company, Touch Press, a nice book by Theodore Gray, who was long time involved with our company, still involved with our company. The most notable in the world, perhaps, for creating the original notebook interface for Wolfram language and so on, back in 1987 or so. But anyway, Theo Gray produced a book about molecules that you can get on the iPad that is rather lovely because it actually has sort of realistic molecules that you can touch with your fingers and pull the atoms apart and see the molecule kind of flopping around as molecules flop around. It's kind of about the only application I've ever seen for the iPad where the fact that you can actually put 10 fingers down and sort of touch 10 different atoms is useful. But in any case, so one of the things you kind of learn from playing around with that is that there are molecules that are really quite rigid and there are molecules that are very, very floppy. And there are molecules where there are sections of the molecule that are very floppy and can twist around and other parts that are quite rigid and so on. But when you have a big biomolecule, and the question is, what does it do when you deform it in some way? Really hard to tell. And so for example, there are efforts to make special purpose computers to do that. A whole friend of mine has built many such things. But it's really hard. And there's a lot of cleverness to do with how you actually do the computations for the molecular dynamics and whether you can do them in parallel, whether you can look at, have different parts of a computer, different computers connected together, where one computer is dealing with the atoms over here in the molecule, another computer is dealing with the atoms over here in a molecule, and run those two computers in parallel, whether you have to sequentially look at first these atoms, then these atoms, then these atoms, and so on. But the question of whether there's a better way to do it, and perhaps using I've seen it before, machine learning type methods, I think is still unclear. It's still sort of an open question. It's quite possible that there will be ways to do that. One problem with the I've seen it before method of saying what happens is, if it turns out you haven't seen it before, you might get a totally wrong answer. If you've got something where it sort of looks kind of like something you've seen before, but look, there's something a little different and you didn't key into that difference properly. People wonder whether you might, maybe I shouldn't get into this particular rabbit hole, but people wonder whether you can use quantum computers to do simulations of quantum molecules and so on. Well, the answer is, almost by definition, the answer is yes. The problem is that to know what the quantum computer did, and this is sort of the nasty underbelly of quantum mechanics. Yes, the thing can inside, it can quantumly figure out what's going on, but us humans think definite things happen in the world. And so all those parallel threads of what might have happened in quantum mechanics, we have to resolve those to actually come out with an answer. And that process of resolution is hard. We don't even know how hard. And so I think that it's not such a great idea to say, oh, we'll just make a quantum computer do it, because yes, the quantum computer is quantum mechanical like the molecule is. And so yes, inside it's all great, but to read out the answer is a whole challenge. So, I mean, Baker asked this question about, is it necessary to have supercomputers to do biomolecule simulations? There are some places where it isn't so necessary. For example, RNA, it folds up in ways that have definite motifs and are computable without a very fancy supercomputer. Proteins have been hard. The dynamics of proteins has also been hard. I think that it could very well be the case that over time, we discover sort of the patterns by which these things work, and we may be able to, rather than having to trace every single atom individually, we may be able to say, oh, we just need to look at this collective thing of 1,000 atoms. We now know what we need to look at about those 1,000 atoms to figure out how they'll interact with another 1,000 atoms. And that may make it much less computation intensive to do these computations. Let's see. Some other things here. Oh boy, a lot of interesting questions here. Aaron asks, what life forms have arbitrary differences between individuals, and what life forms have meaningfully, quotes, unique individuals? It's an interesting question. I think that, well, when you have something like a virus, which has a certain DNA sequence, RNA sequence, like our unfriendly COVID virus has an RNA sequence that's about 29,000 base pairs long. And you say, which particular COVID virus do you have? Well, there are all these variants, all these strains. Within a particular strain, at 29,000 base pairs, you may have an identical sequence of base pairs. But between different strains, you'll see differences in those base pairs. And one might have seen those plots of sort of the genealogy of the viruses, bunch made by a former employee of ours, actually, who's now a well-known virologist, showing kind of the tree by which this sort of divergence of, first there was one virus and then there was a mutation in that virus, a change in the genetic sequence of the virus, and then there were two different viruses that sort of came from that one, and you end up with this whole kind of tree of variants and so on. And there are a great many variants at this point among viruses. So those have sort of within each variant, the viruses may be identical, but between variants they'll be different. So when you have different individuals, so for example, for identical twins, humans, let's say, who have identical DNA, precisely identical DNA. Not every feature of those twins will be identical. The details of their fingerprints, the detailed folding of their ears or their brains or whatever else will be different. It may have some of the same overall features, but some of that detail was the result of features of the development of that human being, and it won't be identical. It's epigenetic. It's something beyond the genetics of that human. And so individuality arises by virtue of sort of the actual growth of the organism, as well as at the level of DNA. And there are some organisms where the placement of cells, if it's a multicellular organism, is always the same. And there's some organisms like us where even between identical twins, the details of where cells are placed will be different. You do a retinal scan of those twins, it'll be a completely different pattern of blood vessels at the back of their eyes, although maybe there's some overall features that were inherited, but the detailed way in which the blood vessels sort of filled out, the capillaries filled out the the back of the eye will be different. There are organisms where that isn't the case, where the cells always form in the same way. So, for example, C. elegans, the nematode, a little tiny worm, it has, oh, how many is it? About 2,000 cells in it. And pretty much every nematode is the same. The cells are arranged in a particular structure, a particular, the mouth cell goes here and the brain cell goes there and so on. And it's precisely determined. But when you get to larger organisms, that's no longer the case. And there'll be features that are that are different between different members of the species. So I'm guessing, I don't know exactly where the dividing line is when the fates of cells and the locations of cells in the organism are precisely determined. It's bigger than 2,000, it's smaller than 1 trillion. My guess is that by the time you're out of the tens of thousands of cells, you're at the point where it's no longer the case, where the overall form of where the cells go may be determined, but the particular cell by cell arrangement is no longer precisely determined. There may be pieces like, I don't know, the motors in a for some kind of flagellum for some little creature, that may be very precise. It almost looks like a crystal. It has its little molecular units arranged so precisely, but there are other parts, and even actually in us, are there places in us where things are arranged kind of at that level of molecular detail? Yeah, I mean, there's certainly things at the level of small clusters of molecules where that's the case. But I think when it comes to the large scale structure of any of our organs or other kinds of systems, I don't think that's the way it works. I don't think it's precisely determined in that way. Well, there's a question here from Anon Emaus. Are Darwin's survival of the fittest, evolution, and machine learning all basically the same thing? If not, how do they differ? Well, so let's see. The question of a lot of these have to do with, you've got a certain set of constraints. How do you find the best way to satisfy those constraints? For example, let's say you are you've got some mountainscape, you've got some collection of hills and valleys and so on, and you're trying to find where's the lowest point in this mountainscape. And you're just walking around, you're an ant on this, or a person on this mountainscape, and you don't have a topographic map or a GPS or a satellite or anything like that. You're just walking around on the mountains trying to find where's the lowest point. And so then the question is, how do you best do that? And that's a problem of optimization. It's a problem of how do you get to the lowest point? And you might say, well, I'm walking down this mountain and I'm down and here. I can look up on all sides of me, things are going up. So I'm at the lowest point. Well, that might not be true, because it might be one valley over, there's a lower point. And but you'd have to go over the mountain to get to that lower point. And if all you're doing is to say, am I at the lowest point? Let me see whether everything from me is up. Then you wouldn't notice that still lower point. And so one approach to finding the lowest point is so-called gradient descent. You say, wherever I am, pick the path that makes me go down the fastest from where I am. And yes, that will have the feature that it will often lead you sort of down the mountain as quickly as possible, but it might lead you to something which is in one valley and not in another valley, and you might miss the lowest valley. You might find the local optimum, but not the global, all over the landscape optimum. So that's a sort of a feature there. But in many of these problems, one's somehow trying to find some kind of optimum, or at least better than the other guy optimum. So in biological evolution, what one's interested in is that one's organism, so to speak, one's species is going to do better if it's somehow better suited to the environment. You know, if it turns out you need to be able to eat those leaves from the top of the tree, the longer your neck is, the more likely you are to be able to eat from the top of the tree and to survive and have baby giraffes or whatever. But the thing you're trying to do ultimately is to optimize things so that you eat best from the top of the tree. So then the question is, what is the best strategy for finding the type of organism that would be able to eat, or the type of thing that would be able to eat from the top of the tree? Well, you could adopt a variety of different strategies. Biological evolution adopts a particular strategy, according to Darwinian evolution. It basically makes lots of variant giraffes, and then each giraffe does its thing, and the giraffes that manage to eat best from the top of the tree will have the most, let's imagine, will have the most little giraffes. And so the trait of being able to eat from the top of the tree, which is passed from giraffe to baby giraffe, so to speak, will tend to win out because one has because the more successful organism, the one that is more optimal, will have more children, and therefore there will be more likelihood that there'll be its genetics, its program will tend to win out against other ones. So that's kind of the approach in Darwinian evolution. You have in parallel lots of little giraffes, and some of them do better than others. In something like machine learning, you're also interested in finding, it's not the construction of the giraffe you're interested in. It's, for example, the construction of a neural net. And you're asked the question, which neural net with its little detailed connections inside it will best recognize cats from dogs? And you want to keep tweaking the neural net until it does the best at recognizing cats from dogs. The strategy that's used there is a little different. The strategy that's used there, the most common one, is backpropagation, where you kind of see, well, how far off are you? And then you kind of go back more in an engineering way and say, well, what do I need to change to make me go sort of more in that direction? So actually, maybe I should make okay, so there's a sort of commonality and difference. I mean, the particular approach of natural selection, of having lots of baby giraffes and so on and seeing which one wins, doesn't tend to be in detail the way that one does things in machine learning. There are methods for doing machine learning which are much more like that, but the most common ones with neural networks, that isn't the most common way to do things. Instead, it's a much more sort of organized, it made the thing go off in this direction. We can figure out how to tweak it so that it doesn't go as much in that direction. It's a more kind of engineering solution. But at the biggest level, at the sort of broadest level, you're still trying to find an optimum. You're trying to find the best neural net that best distinguishes cats from dogs. You're trying to find the best genetics for the giraffe that best eats from the top of the tree, and so on. And in both cases, you're actually making small incremental changes, typically, we don't completely know about biological evolution, but it seems like it's usually the case that the giraffe will have a slightly longer, the child giraffe will have a slightly longer neck, a slightly shorter leg, a slightly different, mouth structure or whatever else. That it seems like biological evolution mostly just makes these small incremental changes. It's kind of almost like that idea, if you're walking around on the mountain, you're making these small moves to get closer to the bottom of the mountain or whatever. We don't completely know whether biological evolution sometimes makes bigger jumps. We can't really see necessarily. We can, in the fossil record, for example, we often will see, well, first there were these kinds of creatures and then there were those kinds of creatures. But actually there was 1 million years between those two types of creatures showing up in the fossil record. We don't completely know what happened in between. And sometimes what happens is there's a gradual change, like a creature that was walking on 4 legs will gradually change its mechanics. And then one day, one of those creatures will realize, gosh, I can walk around on just 2 legs. And then things will rapidly change it. And that's why, so perhaps in the fossil record, it looks as if it went from 4-legged to 2-legged very quickly, even though it had been sort of a gradual process of one set of legs getting longer and shorter, and then suddenly it could become two-legged type thing. And so we don't completely know. We know that in things like viruses, there's some sort of violent changes of genetics where there are crossovers, where sort of pieces of a viral genome will get exchanged with other organisms. A big chunk of genome will get exchanged. That's probably happened with us as well. Between viruses and us, there's probably been a bunch. We know of some examples of that, of exchanges with different organisms with us, where big chunks of genome get changed. But the full story of how that works is really not known. I mean, we can get some idea of that by looking at microorganisms and asking the question. You have a plate, for example, and you have some particular kind of bacteria, and you have an antibiotic, and the antibiotic is killing off those bacteria. But as you go on, the bacteria will evolve. You'll get mutations to the bacteria, and eventually those bacteria might become resistant to the antibiotic. And so then you'll see on this plate a different bacteria. Maybe you can stain them in different colors. Different bacteria will kind of grow past the antibiotic. Then you put a stronger antibiotic on, and then you'll kill off those bacteria. But then eventually a little finger will evolve beyond that, where they evolve to be resistant to that antibiotic, and so on. And I don't know to what extent. I mean, the pictures I've certainly seen of that, it kind of seems like there are sort of occasional discoveries, so to speak, made by the bacteria, occasional kind of jumps where the bacteria sort of discover some feature. They make some new protein or something like that allows them to be resistant to bacteria. But I don't think we really know with clarity what the sort of the gradualness versus jumpiness of biological evolution is really like. Because sometimes the difference between what happens in the program and what happens in the final organism, the genotype in the program versus the phenotype of the final organism, that transformation is a very difficult one. And we don't necessarily know what effect even small changes on the genome will have on the actual form of the organism, and that doesn't help things. So the details of whether, to the extent to which both biological evolution and machine learning and optimization, the extent to which all three of these use sort of gradual incremental change, they all do to some extent. We don't know. In the case of biology, we don't completely know. In the case of optimization of this, like find the minimum on the mountain, there are certain shapes of mountains, certain mathematical forms where vastly more efficient techniques can be used than this kind of just move down the gradient vector and keep going. In fact, that was one of the things in operations research and linear programming, something invented in the 1930s, 1940s, sort of critical to the efficiency of things like factories and so on. It's like, if you're trying to make this number of widgets, how many of this piece of raw material and that piece of raw material do you need? And how does that all sort of fit together? That becomes a problem of linear inequalities. You're taking sums of variables and so on with multiplied by constants. You're saying that's got to be greater than this. We've got to have at least this number of widgets have to be in stock for us to go on and make these other widgets and so on. And so it became of great interest for many kinds of problems to both sort of in terms of sort of supply chain type things, and in terms of things like how do we optimize the routes for an airline, things like this. Those became problems of linear programming, of solving these linear inequalities, and very big systems of linear inequalities, millions sometimes of those. And what happened was that in the 1940s, I guess, methods were invented that were not just, oh, you just incrementally change things until you got a good answer, which actually hadn't been done because things were still being done by hand. And that wasn't a very convenient thing to do by hand. Instead, the so-called simplex method was invented, where it's just like you can just go kind of almost immediately, you go to the sort of corner that optimizes things. And so that was a method that wasn't incremental. And there are other methods of so-called convex optimization, which also aren't incremental like that. So it isn't the case that all optimization happens incrementally, sort of a small step at a time. But some of the ones that seem to happen in nature, for example, that's a convenient trick that nature has learnt. And it's also something which tends to happen in some types of machine learning also. All right, maybe one more, two more here. Architect asks about the formation of buckyballs. So buckyballs are a form of carbon that in which the most common form is carbon-60, in which there are 60 carbon atoms arranged in kind of at the seams of a soccer ball. And the thing that, when you have a bunch of carbon atoms, you can kind of pack them together in different ways. So the two most famous ways are graphite and diamond. In graphite, the carbon atoms are kind of packed in layers. And in diamond, both graphite and diamond are pure carbon, just a bunch of carbon atoms. Diamond, the carbon atoms are arranged in a particular packing pattern that isn't in layers, that is a particular structure, usually called the diamond crystal structure. It's a repeating structure, but it isn't a layered structure. It's a little bit more like the kind of structure you get if you look at how fruit is piled up in a grocery store. It isn't exactly that structure, but it's something similar to that. So those are two forms of carbon that one knew, that have been known for a long time. Question was, are there any other ways to put pure carbon atoms together? Well, it turns out there are. And actually, this wasn't discovered until the 1970s, possibly even 1980s. And it was discovered by pretty fancy techniques involving kind of analyzing the structure of molecules and so on. It's not easy because molecules are really small. You can't just directly look at them. You have to use indirect techniques, using X-rays or using other kinds of techniques. You have to indirectly deduce the structure of molecules, and it's often quite challenging to figure out what they might be. But so in the case of carbon, people wondered, could you have carbon arranged itself in different ways, in ways that weren't just graphite or diamond? And people sort of played around with that, and they managed to make some examples where there were at least carbons and a bunch of hydrogens hanging off them and so on. But then it was discovered that actually in common soot, there are carbons, clusters of carbon atoms that get made that actually form molecules. And they are these molecules that are things like these cages, like the C60 molecule that is a buckyball. A buckyball is a combination of hexagonal faces and pentagonal faces, kind of the way that a soccer ball works. If you had all hexagons, if you just, they'll just form, they'll just tile the plane. It's just like a bathroom tiling or something, where you have a bunch of hexagonal tiles. They just are flat on the plane. If you have pentagons, on the other hand, if you just try and arrange pentagons together, they're not flat at all. You just put a bunch of pentagons together, they will arrange themselves specifically into a dodecahedron, a 12-faced object, 12-faced polyhedron. That's the only way you can put a bunch of pentagons together. But if you mix hexagons and heptagons, hexagons make something flat, pentagons make something curl up. If you have mostly hexagons, but just a few pentagons, then you can make something which has sort of an arbitrary curvature. You can have something where you just have just a very, very few pentagons. It'll be almost flat, but just a little bit curled up. The more pentagons, the more curled up it will be. So you can get different buckyballs that have different kinds of curvature, which are like the less curved, the more flat it is. It's more hexagons, less pentagons, and so on. So buckyballs have this feature that they're actually made in pretty easily in soot and so on. They can be refined in a variety of ways, but they're not so hard to make. Now, people have found that there are different structures of carbon. You can also make graphene, and you can make carbon nanotubes. Carbon nanotube is kind of like a cylinder of carbon, where as you go around the cylinder, you've got these pentagon, hexagon type things, like in a buckyball, but they're arranged to make a cylinder. Carbon nanotubes are pretty interesting. They're very strong, and they're very good electrical conductors. And people talk about weaving them to make things that are strong enough to even be able to lasso a satellite in orbit around the Earth and have like a space elevator where you can go from the surface of the Earth into orbit. I'm not quite sure if that works. Nobody's built one yet, but I'm not quite sure, even if theoretically yet carbon nanotubes are strong enough and sort of spinnable, knit together in such a way that you could do that. But in any case, that's kind of the idea of, and people imagine some things have been made with wires being made from carbon nanotubes and then them crossing over and making gates. And you can sort of imagine making some different form of electrical circuit, which is made sort of more at the molecular scale with just these pieces of carbon and electrons kind of going through at that. And that's kind of a coming attraction of kind of a new form of electronic-like device. Let's see. Oh, I'm just going to pick one more here, and then I really should get going from www, asking how are gemstones formed and how can we model gem features, colors, textures, asterism, anisotropy, everything. Okay, so what is a gemstone? Gemstone is a crystal. What is a crystal? A crystal is a regular arrangement of atoms. And there are, oh my gosh, how many? 16 Bravais lattices, if I remember correctly. There are different ways that you can arrange atoms to make a regular crystal, but there are only a limited number of ways because there are only certain kinds of symmetries that you can have. So for example, just like if you imagine, if you imagine putting together identical objects, let's say you have cubes, you can put together identical cubes and they'll be in a cubic lattice. Let's say you pick some other kind of shape. It's not so easy to find shapes that you can put together identically to form in space, but there are other ones. Let's see, a snub cube is another one, and there are a bunch of these that are not such common shapes. They're not the ordinary polyhedra, so to speak. They're not the ordinary regular platonic solids. There are other kinds of solids, and I'm suddenly slipping my mind. I have a nice picture of these in my book, New Kind of Science, but they don't have particularly common names. But anyway, there are different polyhedra, but you can arrange them in space in a very regular way. So it's just one, every section looks the same, but they don't have the same overall structure as, for example, cubes arranged together. So for example, one thing you can notice is if you have a cube, the cube has 6 faces. That means there are 6 cubes that are adjacent to that cube. But actually, there are other arrangements of things where you, for example, can have 12 things adjacent to a given thing. That's a different structure. Anyways, there are a limited set of these possible crystal structures, and those determine the way that the atoms, and sometimes, so some gemstones, they're not, something like diamond consists just of carbon atoms. Something like quartz has silicon oxide, is that right? It doesn't just have one kind of atom in it. There are other kinds of crystals which can just be copies of one atom, but there are crystals in which there is the unit cell of the crystal, the thing that repeats, contains more than one atom. Another example is table salt. Sodium chloride has sodium and chlorine atoms arranged in that particular case, they're in a cubic arrangement. But so you have this unit cell, which is made of some collection of atoms. And then the crystal is a repetition of that unit cell many, many times. And There are only a limited number of kinds of symmetries of repetitions that you can have, but the unit cells can be of different forms, and different gemstones have different unit cells, and they'll typically have silicon, aluminum, oxygen, sometimes some more obscure metals, iron, manganese, all these kinds of things. And any given type of gem will have a particular set of particular sort of chemical formula, in effect, a particular set of atoms that form its unit cell. Now, it's a little tricky because the internal structure of the crystal will have some particular form that has a particular way of arranging the unit cells. The outer boundary of the crystal may not, has a complicated relationship to what's going on inside. So, for example, for diamonds, it's a big art to cut diamonds in different ways. Whenever you cut a diamond, what you're doing is you're cleaving between atoms. You're cutting through a plane, but between planes of atoms. You don't. It's much harder if you wanted to cut in a diamond. If you are going to sort of you're trying to break between these two atoms and those two atoms in different ways. That's a much harder cut to make than if you're kind of slicing between planes of atoms. But there are many different slicings that you can make between planes of atoms, and different cuts of diamonds correspond to different choices of those slicings. Those slicings are labeled, technically they're called Miller indices. They are labeled by kind of which, sort of how many atoms across do you go for how many atoms down, more or less, in this crystal lattice. And that labels different planes that you can use to slice a crystal on. And so, for example, with diamond, people make up these things, so-called brilliant cuts of diamond, which are particular ways of slicing the diamond so that light, because diamond has a very high refractive index, 2.7, light, when light gets into diamond, it will tend to bounce around inside the diamond. And if you arrange the angles at the outer border of the diamond in just the right way, by cutting it the right way, you'll have many reflections of the light inside the diamond, which makes the diamond sparkle. So in any case, when you see a gemstone that's been cut, it's been cut along these cleavage planes in the crystal, but the details of how it's cut are often part of the story of, that's a cute gemstone type of thing. So the other thing about gemstones is they tend to have, they have imperfections, because when the crystal grew, the atoms were being arranged. Some crystals grow very quickly, some grow slowly. You can have a crystal of, like an alum crystal, sodium thiosulfate, I think, which grows like in moments. You've got the whole crystal. You can have a copper sulfate crystal. I remember growing those when I was a kid. They're nice blue crystals, and you make this this liquid solution of copper sulfate. And as the water kind of evaporates out, you get sort of a higher density of copper sulfate. And if you put a little string in and you seed the crystal, you can, if you do it right, get this big parallel pipette shaped crystal coming out. It's this big blue copper sulfate crystal. And what happened there is the molecules of copper sulfate, gradually attached to the growing surface of the crystal. You got this whole crystal. But when a crystal is grown or exists in the ground or whatever else it's grown in some geological process, often it won't form perfectly in any of these situations. Even in the copper sulfate crystal you grow with a little string and so on, it typically won't form perfectly. There will be little imperfections, either imperfections in the fact that the crystal lattice doesn't align properly. There was a missing molecule or something that caused a sort of a defect, maybe a screw dislocation where there's a sequence of atoms in a kind of helical shape that are missing, other kinds of things like that. Or that are misplaced, rather, I should say. So there are defects in the crystal. There are places where you can actually have an impurity in the crystal, some other substance that got into the crystal as it was forming. So those are things that can happen. The question of in their natural habitat, so to speak, when crystals form, sometimes like quartz crystals, for example, you'll often see these very faceted shapes and so on. But other crystals they'll be kind of messy on the outer boundary will be kind of messy, and only when you cut them will they look really sort of gemstone-like. Another feature of gemstones is their colors, and sometimes the very bright colors of gemstones come from rather interesting sort of phenomenon that there are what they're called color centers, I think F centers, I believe. So normally, what makes the color of something? What makes the color of something is if you start, white light is a combination of all the different colors of the rainbow. And if you, for example, absorb from the white light that, for example, is made by the sun, if you absorb all the blue light, then you'll have red left over. If you absorb all the red light, you'll have blue left over, and so on. And so the question is, what colors of light get absorbed or not by different kinds of crystals? So a diamond is more or less transparent normally. But the question of how it will, and that happens because, well, in a diamond, when a photon of light comes into a diamond, it doesn't, the diamond is sort of letting through all the different energies of photons corresponding to all the different colors, and that's why it's transparent. But what can happen is that a sort of a cage can be created within the crystal in which there is an imperfection, and an atom can be in that imperfection. And what can happen is a photon comes in and it hits that atom in the imperfection, and it causes that atom to be, it makes that atom sort of bounce around. And depending on sort of the size of the imperfection, the type of atom it is, the size of the cage and the type of atom, it can be a particular color of a particular energy of photon, a particular color of light, a particular frequency of light that will excite that atom. And that's why you can have these very, very specific colors of gemstones, has to do with that phenomenon. And that's what can happen there. There's a bit more detail to that, and there's some interesting quantum mechanics and so on that goes along with how that excitation of those atoms works in those cases. But that's that basic phenomenon. I think, gosh, there are so many other things that happen in gemstones. I mean, another common phenomenon is what's called birefringence. We talked about light, different colors of light getting through or not getting through, leading to different, leading to the appearance of different colors of the gemstone. But another thing that can happen, light is polarized. So it can be polarized in the sense that light consists of, you can think of it as sort of electric field oscillations and magnetic field oscillations. They're at 90 degrees to each other. And you can have it be the case that the light is oriented so that the electric field is going one way or the electric field is going the other way. And those two different orientations can be treated differently by a crystal. So for example, calcite is a common kind of crystal that has this phenomenon that light polarized one way goes at a different speed in a calcite crystal from light polarized the other way. And so that means that, in fact, you end up getting two images often in a calcite crystal, because those light going different speeds means that the refractive index is different, the light is bent differently. So that means you put a calcite crystal, you look through a calcite crystal, and you'll see a double image of the thing underneath it, because the different polarizations of light were bent at different angles as they went through the crystal. Comment here by Parker, quartz, they notice imperfections like streaks that are kind of molecular analogues of cellular automata. I don't know specifically about that. I have looked quite a bit at crystal growth because it is a good example of systems that grow according to simple rules. I've looked particularly at dendritic crystal growth, like snowflakes with arms and so on. Streaking, I'm familiar with the phenomenon, but I don't know what a good model for it. Interesting thing to look at. All right. Well, lots of interesting questions today. Thank you all very much for that. And I should sign off here and see you another time. Thanks for